The activated transducin subunit splits away and activates PDD. 14 light absorption closes sodium channels. The absorption of light by rhodopsin initiates a cascade of events resulting in the hyperpolarization of the rod cell. L activated PDD. Hydrolyzes cGMP to 5-GMP, causing NOT channels to close. Cytoplasm of disc. Arthropods have evolved compound eyes that provide them with information about patterns or images in the environment. Each compound eye consists of many optical units called ommatidia, singular, ommatidium figure 15. The number of ommatidia in a compound eye varies from only a few in some ants to 800 in fruit. Flies, to 10,000 in some dragonflies. A corneal lens. The compound eyes of a fruit fly each contain. Dot. Hundreds of ommatidia, ommatidium bundle of axons. To brain. Basement membrane. 15 ommatidia, the functional units of insect eyes. Up the micrograph shows the compound eye of a fruit. Fly Drosophila. Beat the rhodopsin-containing retinal cells are the photoreceptors in ommatidia. Each ommatidium has a lens structure that directs light onto photoreceptors. Flies, for example, have 8 elongated photoreceptors in each ommatidium. The inner borders of the photoreceptors are covered with microvilli that contain rhodopsin and trap light. Axons from the photoreceptors communicate with the nervous system. Since each ommatidium of a compound eye is directed at a slightly different part of the visual world, only a crude, or perhaps a broken up, image can be communicated from the compound eye to the CNS. Image-forming eyes evolved vertebrates and cephalopods. Independently in. Both vertebrates and cephalopod mollusks have evolved eyes with exceptional abilities to form images of the visual world. Like cameras, these eyes focus images on a surface that is sensitive to light. Figure 16. Considering that they evolved independently of each other, their high degree of similarity is remarkable. A camera's lens focuses an inverted image on the film in the same way the eye's lens focuses an image on the retina. Ciliary muscle. Suspensory ligaments, optic nerve. Blind spot. Fovea. Retina. Iris T sclera. The eye of the squid is 16 eyes like cameras very similar in structure to the vertebrate eye, butted lens. Cornea, 8, evolved independently. Iris optic nerve. Equals double layer of receptor cells. Eye of squid. The vertebrate eye once a spherical, fluid-filled structure bounded by a tough connective tissue layer called the sclera. At the front of the eye, the sclera forms a transparent cornea, through which light passes to enter the eye. Just inside the cornea is a pigmented iris, which gives the eye its color. The function of the iris is 80 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. To control the amount of light that reaches the photoreceptor cells at the back of the eye, just as the diaphragm of the camera controls the amount of light reaching the film. The central opening of the iris is the pupil. The iris is under neuronal control. In bright light, the iris constricts, and the pupil is very small. As light levels fall, the iris relaxes, and the pupil enlarges. Behind the iris is a crystalline protein lens, which makes fine adjustments in the focus of images on the photosensitive layer, the retina, at the back of the eye. The most sensitive area of the retina is called the fovea. The cornea and the fluids within the eye are mostly responsible for focusing light on the retina, but the lens allows the eye to accommodate, that is, to focus on objects at various locations in the near visual field. To focus a camera on objects close at hand, you adjust the distance between the lens and the film. Fishes, amphibians, and reptiles accommodate in a similar manner, moving the lenses of their eyes closer or farther from their retinas. Mammals and birds use a different method. They alter the shape of the lens. The lens is contained in a connective tissue sheath that tends to keep it in a spherical shape, but it is attached to suspensory ligaments that pull it into a flatter shape. Circular muscles called the ciliary muscles counteract the pull of the suspensory ligaments and permit the lens to round up. When the ciliary muscles are at rest, the flatter lens has the correct optical properties to focus distant images on the retina, but not close images. Contracting the ciliary muscles rounds up the lens, changing its light-bending properties to bring close images into focus. Figure 17. As we age, our lenses become less elastic, and we lose the ability to focus on objects close at hand without the help of corrective lenses. As a consequence, most adults over the age of 45 need the assistance of bifocal lenses or reading glasses to compensate for their lost ability to accommodate. The vertebrate retina receives and processes visual information. During embryonic development, neuronal tissue grows out from the brain to form the retina. In addition to a layer of photoreceptor cells, the retina includes 4 layers of cells that process visual information from the photoreceptors and produce an output signal that is transmitted to the brain via the optic nerve. The light-absorbing outer segments of the photoreceptor cells are all the way at the back of the retina. Light must pass through all the layers of retinal cells before being captured by rhodopsin. The outer segments are partly buried in a layer of pigmented epithelium that absorbs photons not captured by rhodopsin and prevents any backscattering of light that might decrease visual sharpness. The photoreceptors of the retina, intel. Now we have referred to only one kind of photoreceptor cell, the rod cell. But there are 2 major kinds of vertebrate photoreceptors, both named for their shapes, rod cells and cone cells. Figure 18. A human retina has about 5 million cones and about 100 million rods. Rod cells are highly sensitive to light, so they are well suited for vision under dim light or saturated by daylight conditions and do not contribute to color vision. Cone cells are less sensitive to light, so they are better suited for daylight and color vision. Cones are also responsible for our sharpest vision. Even though there are many more rods than cones in human retinas, our foveas contain only cones. For near vision, ciliary muscles contract, causing the lens to round up. Suspensory ligaments. For distant vision, ciliary muscles relax and suspensory ligaments pull lens into a flatter shape. Opening square bracket. 17 staying in focus. Mammals and birds focus their eyes by changing the shape of the lens. Rod cells, cone cells. 18 rods and cones. This scanning electron micrograph of photoreceptors in the retina of a mud puppy and amphibian shows cylindrical rods and tapered cones. Because cones have low sensitivity to light, they are of no use in dim light. At night our vision is not very sharp, and we see mostly in shades of gray. You may have trouble seeing a small object such as a keyhole at night when you are looking straight at it, that is, when its image is falling on your fovea. If you look a little to the side, so that the image falls on a rod-rich area of your retina, you can see the object better. Astronomers looking for faint objects in the sky learned this trick a long time ago. Animals that are nocturnal, such as flying squirrels, have retinas containing a high percentage of rods and may have poor color vision. By contrast, some animals that are active only during the day, such as chipmunks, have mostly cones in their retinas. The human retina has 3 kinds of cone cells, each containing slightly different isomers of opsin. These opsin molecules differ in the wavelengths of light they absorb best. Although the same 11 cis retinal group is a light absorber in all 3 kinds of cones. See figure. Institute for Advanced Studies, Jodhpur 81. Unit 7 System Physiology Animal. 12 Its molecular interactions with opsin determine the spectral sensitivity of the rhodopsin molecule as a whole. One isomer of opsin causes the retinal group to absorb short wavelength light, e.g., violet and blue most efficiently. The others result in absorption of middle wavelengths, e.g., green long wavelengths, e.g., yellow and red. Figure 19. RELAT versus E. ABSOR BA NCD. 400,450. Wavelength NM. 19 absorption spectra of cone cells. The 3 kinds of cone cells contain slightly different opsin molecules, which absorb different wavelengths of light. The density of rods and cones is not the same across the entire retina. In humans, light coming from the center of the visual field falls on the fovea, where the density of cone cells is highest. The human fovea has about 160,000 photoreceptors per square millimeter. A hawk has about 1 million photoreceptors per square millimeter, making its vision sharper than ours. In addition, the hawk has 2 foveas in each eye. One receives light from straight ahead, while the other receives light from below. Thus, while the hawk is flying, it sees both its projected flight path and the ground below, where it might detect a mouse scurrying in the grass. Where blood vessels in the optic nerve pass through the back of the eye, there are no photoreceptors, so there is a blind spot on the retina. See figure 16. You are normally not aware of your blind spot, but you can find it. Stare straight ahead, holding a pencil in your outstretched hand so that the eraser is in the center of your field of vision. While continuing to stare straight ahead, slowly move the pencil to the side until the eraser disappears. When this happens, the light from the eraser is focused directly on your blind spot. Information flow in the retina. The human retina is organized into 5 layers of neurons that receive visual information and process it before sending it to the brain. Figure 20. A first step in understanding how the retina tells the brain what it is seen wants to study how these layers are interconnected and how they influence one another. As we know, the photoreceptor cells at the back of the retina hyperpolarize in response to light and do not generate action potentials. The cells at the front of the retina are ganglion cells. They fire action potentials, and their axons form the optic nerve that travels to the brain. The corneal lens. Light. 20. The retina. The human retina has 5 layers of neurons that receive and process visual information. Layers of cells between the photoreceptors and the ganglion cells process information about the visual field. The photoreceptors and ganglion cells are connected to one another by bipolar cells. Changes in the membrane potential of rods and cones in response to light alter the rates at which the rods and cones release neurotransmitter at their synapses with the bipolar cells. In response to neurotransmitter from the photoreceptors, the membrane potentials of the bipolar cells change, altering the rate at which they release neurotransmitter onto ganglion cells. The rate of neurotransmitter release from the bipolar cells determines the rate at which the ganglion cells fire action potentials. Thus, the direct flow of information in the retina is from photoreceptor to bipolar cell to ganglion cell. The ganglion cells send the information to the brain. The other 2 cell layers, the horizontal cells and the omocrine cells, communicate laterally across the retina. Horizontal cells form synapses with neighboring photoreceptors. Thus, light falling on one photoreceptor can influence the sensitivity of its neighbors to light. This lateral flow of information enables the retina to sharpen the perception of contrast between light and dark. Patterns. Omocrine cells form local interconnections between bipolar cells and ganglion cells. The role of omocrine cells is still not entirely understood. Some omocrine cell types are highly sensitive to changing illumination or to motion. Others assist in adjusting the sensitivity of the eyes according to the overall level of light falling on the retina. When background light levels change, omocrine cell connections to the ganglion cells help the ganglion cells remain sensitive to temporal changes in stimulation. Thus, even with large changes in background illumination, the eyes are sensitive to smaller, more rapid changes in the pattern of light falling on the retina. Neurons, ganglion, omocrine, bipolar. Our light travels through layers of transparent and is absorbed by the rods and cones, the photoreceptive layer at the back of the retina, and horizontal cells. A retina to optic nerve 4i. Omocrine. Cell. Bipolar cell. Light. Ganglion cells. Photoreceptor. Cone rod pigmented horizontal cell. Epithelium. Axons of ganglion cells. Visual information is processed through several layers of neurons. Dot and finally converges on ganglion cells, which send their axons to the brain. Information processing in the retina. Knowing the path of information flow through the retina still does not tell us how the information is processed. What does the eye tell the brain in response to a pattern? 82 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Of light falling on the retina, one aspect of information processing in the retina is convergence of information. There are more than 100 million photoreceptors in each retina, but only about 1 million ganglion cells sending messages to the brain. How is the information from all those photoreceptors integrated by the ganglion cells? This question was addressed in some elegant, classic experiments in which electrodes were used to record the activity of single ganglion cells in living animals while their retinas were stimulated with spots of light. These studies revealed that each ganglion cell has a well-defined receptive field that consists of a specific group of photoreceptor cells. Stimulating these photoreceptors with light activates the ganglion cell, figure 21. Information from many photoreceptor cells is integrated in this way to produce a single message to the brain. Experiment. Question: how do retinal ganglion cells code patterns of light falling on the retina? Method. Dash. Light stimulates a small, circular area of the retina. Photoreceptors in a circular receptive field send input to a single ganglion cell. LT photoreceptors. Light. 9 feet underscore. N. 1 an oscilloscope shows underscore action potentials generated by the ganglion cell. Electrode. To visual cortex. Pattern of light bipolar cells stimulating retina. Results. Stimulus patterns on retina. Action potentials in complete darkness. Small spot falling on center of receptive feld. Large spot covering receptive feld. Ring of light excluding center of receptive field. 21 What does the eye tell the brain when the retina is stimulated with dots and rings of light? Individual ganglion stimulus. On center ganglion cell. Of brain. Ganglion cell. Action potentials in off-center ganglion cells show different responses. An on-center ganglion cell is on its receptive field surround. Inhibited by airing of light falling, an off-center ganglion cell is stimulated by light falling on its receptive field surround. And is inhibited by light falling on its center. Conclusion. 1. Ganglion cells have circular receptive fields that are divided into center and surround areas. 2. Some ganglion cells are maximally stimulated by light falling on the center of their receptive fields. Others are maximally stimulated by light falling on the surround of their receptive fields. 3. Ganglion cells encode patterns of contrast between light and dark. The receptive fields of many ganglion cells are circular, but the way a spot of light influences the activity of a ganglion cell depends on where in the receptive field it falls. The receptive field of a ganglion cell can be divided into 2 concentric areas, called the center and the surround. There are 2 kinds of receptive fields, on-center and off-center. Stimulating the center of an on-center receptive field excites the ganglion cell, and stimulating the surround inhibits it. Stimulating the center of an off-center receptive field inhibits the ganglion cell, and stimulating the surround excites it. Center effects are always stronger than surround effects. The response of a ganglion cell to stimulation of the center of its receptive field depends on how much of the surround once also stimulated. A small dot of light directly on the center has a maximal effect. A bar of light hitting the center and parts of the surround has less of an effect, and a large, uniform patch of light falling equally on center and surround has very little effect. Ganglion cells thus communicate information about contrast between light and dark that fall on different regions of the receptive fields. Institute for Advanced Studies, Jodhpur 83. Unit 7 System Physiology Animal. How are receptive fields related to the connections among the neurons of the retina? The photoreceptors in the center of the receptive field of a ganglion cell are connected to that ganglion cell by bipolar cells. The photoreceptors in the surround modify the communication between the center photoreceptors and their bipolar cells through the lateral connections of horizontal cells. Thus, the receptive field of a ganglion cell consists of a pattern of synapses among photoreceptors, horizontal cells, bipolar cells, and ganglion cells. The receptive fields of neighboring ganglion cells can overlap greatly. A given photoreceptor can be effectively connected to several ganglion cells. Thus, the ganglion cells send simple messages to the brain about the pattern of light intensities falling on small, circular patches of retina. Sensory worlds beyond human experience. Humans make use of only a subset of the information available to us in the environment. Other animals have sensory systems that enable them to use different subsets and different types of information. Some species can see infrared and ultraviolet light. When discussing vision, we use the term visible light, but what we really mean is light visible to humans. Our visible spectrum is a very narrow region of the entire continuous range of electromagnetic radiation in the environment. We cannot see ultraviolet radiation, for example, but many other animals can. One of the 8 photoreceptors in each ommatidium of a fruit fly is sensitive to ultraviolet light. The visual sensitivity of many pollinating insects includes the ultraviolet part of the spectrum. Some flowers have patterns that are invisible to us, but show up if we photograph them with film that is sensitive to ultraviolet light. Those patterns provide information to prospective pollinators, but humans are not equipped to receive that information, though some other mammalian species, such as mice, are. At the other end of the spectrum is infrared radiation, which we sense as heat. Other animals extract much more information from infrared radiation, especially that emitted by potential prey. Pit vipers such as rattlesnakes have pit organs, one just in front of each eye, that use highly sensitive heat detectors and a simple pinhole camera arrangement to sense and locate infrared radiation. In total darkness, these snakes can locate a mouse, orient to it, and strike it with great accuracy. A researcher observing elephants had the sensation that there was a throbbing in the air around her. When she obtained microphones and recorders capable of picking up very low frequencies of sound, she discovered that the elephants were communicating with sounds below the range of human hearing. The advantage of using low frequency sound is that it carries over very long distances, so elephants in the wild can communicate even when very far apart. Echolocation is sensing the world through reflected sound. Some species emit intense sounds that are above the range of human hearing, and they use the echoes from those sounds to create images of their environments. Bats, porpoises, and dolphins are all accomplished echolocators. Some species of bats have elaborate modifications of their noses to direct the sounds they emit, as well as impressive ear pinnae to collect the returning echoes. Although we cannot hear them, the high-frequency sounds they emit as pulses, about 20 to 80 per second, are extremely loud in contrast to the resulting faint echoes bound. Some species can see infrared and ultraviolet light. When discussing vision, we use the term visible light, but what we really mean is light visible to humans. Our visible spectrum is a very narrow region of the entire continuous range of electromagnetic radiation in the environment. We cannot see ultraviolet radiation, for example, but many other animals can. One of the 8 photoreceptors in each ommatidium of a fruit fly is sensitive to ultraviolet light. The visual sensitivity of many pollinating insects includes the ultraviolet part of the spectrum. Some flowers have patterns that are invisible to us, but show up if we photograph them with film that is sensitive to ultraviolet light. Those patterns provide information to prospective pollinators, but humans are not equipped to receive that information, though some other mammalian species, such as mice, are. At the other end of the spectrum is infrared radiation, which we sense as heat. Other animals extract much more information from infrared radiation, especially that emitted by potential prey. Pit vipers such as rattlesnakes have pit organs, one just in front of each eye, that use highly sensitive heat detectors and a simple pinhole camera arrangement to sense and locate infrared radiation. In total darkness, these snakes can locate a mouse, orient to it, and strike it with great accuracy. Elephants can communicate with infrasound. Just as there are wavelengths of light beyond our visual capabilities, there are frequencies of sound that we cannot hear. A researcher observing elephants had a sensation that there was a throbbing in the air around her. When she obtained microphones and recorders capable of picking up very low frequencies of sound, she discovered that the elephants were communicating with sounds below the range of human hearing. The advantage of using low frequency sound is that it carries over very long distances, so elephants in the wild can communicate even when very far apart. Echolocation is sensing the world through reflected sound. Some species emit intense sounds that are above the range of human hearing, and they use the echoes from those sounds to create images of their environments. Bats, porpoises, and dolphins are all accomplished echolocators. Some species of bats have elaborate modifications of their noses to direct the sounds they emit, as well as impressive ear pinnae to collect the returning echoes. Although we cannot hear them, the high-frequency sounds they emit as pulses, about 20 to 80 per second, are extremely loud in contrast to the resulting faint echoes bouncing off small insects. An echolocating bat is similar to a construction worker who is trying to overhear a whispered conversation on a street corner while using a pneumatic drill. To avoid deafening themselves, bats use muscles in their middle ears to dampen their sensitivity while they emit sounds, then relax them quickly enough to hear the echoes. The ability of bats to use echolocation to sense their environment is so good that in a totally dark room strung with fine wires, they can capture tiny flying insects while navigating around the wires. Some fish can sense electric fields. The lateral lines of some fish species, such as sharks and rays, contain electroreceptors as well as mechanoreceptors. These sensory cells enable the fish to detect weak electric fields, which can help them locate prey. The use of electroreceptors is quite sophisticated in species called electric fishes. These fishes have evolved electric organs in their tails that generate a continuous series of electric pulses, creating a weak electric field around their bodies. Any objects in the environment, such as rocks, plants, or other fish, disrupt the electric field, and the electroreceptors of the lateral line detect those disruptions. Such electroreceptors allow electric fishes to find food, avoid obstacles, and locate mates in extremely murky waters, such as those of the Amazon River. Electroreception can also play a role in social interactions. In some electric fish species, each individual in a group emits its electric pulses at a different frequency. If a new fish is added to the group, they all readjust their frequencies. 84 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. F. Excretory system. Comparative physiology of excretion, kidney, urine formation, urine concentration, waste elimination, micturition, regulation of water balance, blood volume, blood pressure, electrolyte balance, acid-base balance, tissue fluid and water balance. Life evolved in the seas, and seawater is the extracellular environment for the cells of the simplest marine animals. More complex marine animals have an internal environment consisting of tissue fluid, extracellular fluid, which is isolated from seawater but is similar to it in composition and solute concentration. Most marine and all freshwater vertebrates and terrestrial animals maintain tissue fluids whose concentration and composition differ considerably from that of seawater. The concentration of the tissue fluid determines the water balance of the cells, and the composition of the tissue fluid influences the health and functions of the cells it bathes. Recall, for example, the importance of ionic concentration gradients between the tissue fluid and the cytoplasm of nerve and muscle cells. To understand what is meant by water balance, remember that cell plasma membranes are permeable to water and that the movement of water across membranes depends on differences in solute concentration. Recall the principle you learned in plant physiology. In the discussion of the movement of water in plants, the greater the solute concentration of a solution, the greater the tendency of water to move into it from another solution of lower solute concentration. Likewise, if the solute concentration of the tissue fluid surrounding animal cells is less than that of the cytoplasm, water moves into the cells, causing them to swell and possibly burst. If the solute concentration of the tissue fluid is greater than that of the cytoplasm, the cells lose water and shrink. The solute concentration of the tissue fluid determines both the volume and the solute concentration of the cells themselves. Animal physiologists use the term osmolarity in discussing osmotic phenomena. The osmolarity of a solution is the moles of osmotically active particles per liter of solvent. Thus, a 1 molar solution of glucose is also a 1 osmolar solution, but a 1 molar solution of sodium chloride NaCl is a 2 osmolar solution because each NaCl molecule dissociates into 2 osmotically active particles. To achieve water balance, animals must be able to maintain the osmolarity of their tissue fluid within an appropriate range. In addition, they must maintain an appropriate solute composition of the tissue fluid, saving some substances and eliminating others. To accommodate these needs, most animals have excretory organs. Excretory organs control the osmolarity of tissue fluid by filtration, secretion, and resorption. Excretory organs control the osmolarity and the volume of blood and tissue fluid by excreting solutes that are present in excess, such as NaCl when we eat lots of salty food, and conserving solutes that are valuable or in short supply, such as glucose and amino acids. In terrestrial organisms, these excretory organs also eliminate the waste products of nitrogen metabolism. The output of the excretory organs is called urine. The same basic principles apply to the excretory organs of many species. In some way, the excretory organ filters tissue fluid to produce a filtrate that contains no cells or large molecules, such as proteins. The composition of this filtrate is then modified to produce urine. The filtration process is usually carried out on blood plasma driven across capillary walls in the excretory organ by blood pressure. Water and small molecular weight solutes cross the capillary wall, but large molecular weight solutes and cells remain in the blood. The filtrate water and small molecules then enters a system of tubules. The cells of the tubules change the composition of the filtrate by active secretion and resorption of specific solute molecules. We will see that these three mechanisms, filtration, secretion, and resorption, are used in the excretory systems of a wide variety of animals. The functions of the excretory organs of a species, and therefore the composition of its urine, depend on the environment in which it lives. We will examine the different excretory systems that maintain salt and water balance and eliminate nitrogen in marine, freshwater, and terrestrial habitats. As we shall see, in spite of the evolutionary diversity of the anatomical and physiological details, all these systems obey a common rule. There is no active transport of water. Water must be moved either by pressure or by a difference in osmolarity. Environments and animals can be classified in terms of salts and water. The salt concentration, or osmolarity, of ocean water is about 1070 milliosmoles per liter mosm 1, and fresh water is generally between 1 and 10 mosm L. Aquatic environments grade continuously from fresh to extremely salty. Consider a place where a river enters the sea through a bay or a marsh. Aquatic environments within that Bayer Marsh range in osmolarity from that of the freshwater of the river to that of the open sea. Evaporating tide pools can reach an even greater osmolarity than seawater. Animals live in all these environments. Some aquatic species are osmoconformers. They allow the osmolarity of their tissue fluid to equilibrate with their environment. In contrast, osmoregulators maintain the osmolarity of their tissue fluid at a constant level as the environment changes. OSMOCONFORMERS. Over a wide range of environmental osmolarities, marine invertebrates simply equilibrate the osmolarity of their tissue fluid with that of the environment. There are limits to osmoconformity, however. No animal could survive if its tissue fluid had the osmolarity of fresh water, nor could animals survive with internal osmolarities as high as those that may be reached in an evaporating tide pool. Such solute concentrations cause proteins to denature. Institute for Advanced Studies, Jodhpur 85. Unit 7 System Physiology Animal. OSMOREGULATORS. All animals have solutes in their tissue fluids. Therefore, in freshwater, osmosis causes water to invade their bodies, so osmoregulation is essential. To osmoregulate in freshwater, animals must excrete water and conserve solutes, hence they produce large amounts of dilute urine. In salt water, the opposite problem exists. For animals that maintain the osmolarity of their tissue fluids below that of the sea, osmosis causes a loss of water. To osmoregulate in salt water, animals must conserve water and excrete salts. Thus they tend to produce small amounts of urine, and they have means of excreting salts. Even animals that osmoconform over a wide range of environmental osmolarities must osmoregulate in extreme environments. The brine shrimp Artemia, figure 1, can live in environments of almost any salinity. Artemia are found in huge numbers in the most saline environments known, such as Great Salt Lake in Utah, and coastal evaporation ponds where salt is concentrated for commercial purposes. The osmolarity of such water reaches 2,500 mosm L. At these high environmental osmolarities, Artemia is capable of maintaining its tissue fluid osmolarity considerably below that of the environment, and therefore acts as a hyposmotic regulator. Very few organisms can survive in the crystallizing brine in which Artemia thrives. The main mechanism this small crustacean uses for hyposmoregulation is the active transport of C from its tissue fluid out across its gill membranes to the environment. Ions follow. Artemia cannot survive in fresh water, but it can live in dilute seawater, in which it maintains the osmolarity of its tissue fluid above that of the environment. Under these conditions, Artemia behaves as a hyperosmotic regulator. That is, it maintains the osmolarity of its tissue fluid above the osmolarity of the environment. It achieves this by reversing the direction of C ion transport across its gill membranes. Ionic conformers and regulators. Osmoconformers can also be ionic conformers, allowing the ionic composition, as well as the osmolarity, of their tissue fluid to match that of the environment. Most osmoconformers, however, are ionic regulators to some degree. They employ active transport mechanisms to maintain specific ions in their tissue fluid at concentrations different from those in the environment. The terrestrial environment presents problems of osmoregulation and ionic regulation that are entirely different from those faced by aquatic organisms. Because the terrestrial environment is extremely desiccating, drying, most terrestrial animals must conserve water. Exceptions are animals such as muskrats and beavers that spend most of their time in fresh water. Terrestrial animals obtain their salts from food, but plants generally have low concentrations of sodium, so most herbivores must conserve sodium ions or obtain them elsewhere. Some terrestrial herbivores travel long distances to naturally occurring salt licks. By contrast, birds that feed on marine animals must excrete the excess of sodium they ingest with their food. Their nasal salt glands excrete a concentrated solution of NaCl via a duct that empties into the nasal cavity. Birds, such as penguins and seagulls, that have nasal salt glands can be seen frequently sneezing or shaking their heads to get rid of the very salty droplets excreted from their nasal salt glands. Figure 2. A brine shrimp in dilute seawater actively transports ions into its body to keep the osmolarity of its tissue fluid above that of the environment. But over a wide range of seawater concentrations, it allows the osmolarity of its tissue fluid to equilibrate with the environment. Two nasal salt glands excrete excess salt OSMOLARITY. In highly saline water, the brine shrimp actively transports ions out of its body to maintain the osmolarity of its tissue fluid below that of the environment. Evaporating. Dilute seawater salt pond seawater O equals molarity of environment. One environments can vary greatly in salt concentration. Animals such as the brine shrimp that live at the extremes of environmental osmolarities display flexible osmoregulatory abilities. They become hypersmotic regulators in very dilute water and hyposmotic regulators in very saline water. Salt glands are located in depressions in the skull above the eyes. Ions are carried from the blood to secretory tubules via active transport. C ions follow. The functional unit of a salt gland consists of secretory tubules that drain into a central canal. Nasal salt glands adapted to excrete the excess salt from the seawater they consume with their food. XX spreading nitrogenous wastes. In addition to maintaining salt and water balance, animals must eliminate the waste products of metabolism from their tissue fluid. The end products of the metabolism are carbohydrates and fats are water and carbon dioxide, which are not difficult to eliminate. Proteins and nucleic acids, however, contain nitrogen, so their metabolism produces nitrogenous wastes in addition to water and carbon dioxide. The most common nitrogenous waste is ammonia NH winking phase, which is highly toxic to animals. Ammonia must be excreted continuously to prevent its accumulation, or it must be detoxified by conversion into other molecules before it is excreted. Those molecules are principally urea and uric acid. Figure 3. 86 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Carbohydrates O fats TH, O3 waste products of metabolism. The metabolism of proteins and nucleic acids produces the nitrogenous wastes ammonia, uric acid, and urea. Most aquatic animals, including most fishes, excrete nitrogenous wastes as ammonia. Most terrestrial animals excrete either urea or uric acid. Urea is more soluble in water and is a major nitrogenous excretory product for mammals and amphibians, as well as some fishes. Uric acid is not very soluble in water and is a major nitrogenous excretory product for birds, reptiles, and insects. Aquatic animals excrete ammonia. Continuous excretion of ammonia is relatively simple for aquatic animals. Ammonia is highly soluble in water and diffuses rapidly. Animals that breathe water continuously lose ammonia from their blood to the environment by diffusion across their gill membranes. Animals that excrete ammonia, such as aquatic invertebrates and bony fishes, are said to be aminotelic. Many terrestrial animals and some fishes excrete urea. Ammonia is a dangerous metabolite for terrestrial animals that have limited access to water. In mammals, ammonia is lethal when it reaches a concentration of only 5 milligrams, 100 milliliters of blood. Therefore, terrestrial and some aquatic animals convert ammonia into either urea or uric acid. Ureotelic animals, such as mammals, amphibians, and cartilaginous fishes, sharks and rays, excrete urea as their principal nitrogenous waste product. Urea is quite soluble in water, but excretion of urea solutions at low concentrations could result in a large loss of water that many terrestrial animals can ill afford. As we will see later in this chapter, mammals have evolved excretory systems that conserve water by producing concentrated urea solutions. The cartilaginous fishes are another story. These marine species keep their body fluids almost isotonic to the marine environment by retaining high concentrations of urea. Some terrestrial animals excrete uric acid. Animals that conserve water by excreting nitrogenous wastes as uric acid are said to be uricotelic. Insects, reptiles, birds, and some amphibians are uricotelic. Uric acid is very insoluble in water and is excreted as a semisolid. For example, it is a whitish material in bird droppings. Therefore, a uricotelic animal loses very little water as it disposes of its nitrogenous wastes. Most species produce more than one nitrogenous waste. Humans are ureotelic, yet we also excrete uric acid and ammonia. The uric acid in human urine comes largely from the metabolism of nucleic acids and caffeine. In the 3 == condition known as gout, uric acid levels in the tissue fluid increase, and uric acid precipitates out of solution in the joints and elsewhere, causing swelling and pain. The excretion of ammonia is an important mechanism for regulating the pH of the tissue fluid. In some species, different developmental stages live in different habitats and have different forms of nitrogen excretion. The tadpoles of frogs and toads, for example, excrete ammonia across their gill membranes, but when they develop into adult frogs or toads, they generally excrete urea. Some adult frogs and toads that live in arid habitats excrete uric acid. These examples show the considerable evolutionary flexibility in the excretion of nitrogenous wastes. The diverse excretory systems of invertebrates. Freshwater and terrestrial invertebrates have a wide variety of adaptations for maintaining salt and water balance and excreting nitrogen. In this section, we will explore 3 examples of invertebrate excretory systems: Protonephridia, Metanephridia, and Malpighian tubules. The Protonephridia of flatworms excrete water and conserve salts. Many flatworms, such as Planaria, live in fresh water. These animals excrete water through an elaborate network of tubules running throughout their bodies. The tubules end in flame cells, so called because each cell has a tuft of cilia projecting into the tubule. The beating of the cilia gave the appearance of a flickering flame, figure 4. A flame cell and a tubule together form a protonephridium plural, Protonephridia, from the Greek prota, before, and nephros, kidney. Tissue fluid enters the tubules. How it does so is not entirely clear, and the beating of the cilia causes this fluid to flow through the tubules toward the animal's excretory pore. As it flows, the cells of the tubules modify the composition of the fluid. As the modified tubule fluid urine leaves the flatworm's body, it is less concentrated than the animal's tissue fluid, so ions are conserved and water is excreted by the protonephridium. Institute for Advanced Studies, Jodhpur 87. Unit 7 System Physiology Animal. Greek meta, akinta, and nephros, kidney. Each metanephridium begins in one segment as a ciliated, funnel-like opening in the coelom, called the nephrostome, which leads into a tubule in the next segment. The tubule ends in a pore, called the nephridiopore, that opens to the excretory canal outside of the animal, figure 5. Coulombic fluid enters the metanephridia through the nephrostomes. As the fluid passes through the tubules, their cells actively resort certain molecules from it and actively secrete other tubule cells molecules into it. What leaves the animal through the nephridiopores is a hypotonic dilute urine-containing nitrogenous wastes, among other solutes. Flame cell. Nucleus. The Malpighian tubules of insects depend on active transport. Insects can excrete nitrogenous wastes with very little loss of water. Therefore, some insect species can live in ONIRRTET, BESL, the driest habitats on Earth. The insect excretory system body fluids enter the space enclosed toward the excretory pore by the by flame cell, though consists of Malpighian tubules. An individual insect has beating of the cilia in the flame cell. For Protonephridia in flatworms, the Protonephridia of the flatworm Planaria consists of tubules ending in flame cells. The tubule cells modify the composition of the fluid passing through them. The uric acid precipitates in G. Rectum is the metanephridia of annelids process coulombic fluid, neat section. Tilde secreted. Filtration of body fluids and modification of urine by tubules are highly developed processes in annelid worms, such as the earthworm. Recall that annelids are segmented and have a fluid-filled body cavity, called the coelom, in each segment. Annelids have a closed circulatory system through which blood is pumped under pressure. The pressure causes the blood to be filtered across the thin, permeable capillary walls into the semisolid wastes. KHO0, including uric acid. ANA and K are actively transported from the hindgut and rectum back to the coulombic fluid. H0 follows. Coelom. The cells and large protein molecules of the rectum blood stay behind in the capillaries, while water and hindgut small molecules leave them and enter the coelom. In 52 phi, section, FID, 7G, N2, section P, MFL addition, some waste products, such as ammonia, diffuse ERO, directly from the tissues into the coelom. But where do 6 Malpighian tubules in insects the blind, thin-walled Malpighian tubules are attached to the junction of the insect's midgut and hindgut and project into the spaces containing tissue fluid. This coulombic fluid go from 2 to more than 100 of these tubules. 5 LTGG attached to the gut between the midgut and hindgut. B. Metanephridium alter the W composition of coulombic fluid. Tubules are flow, edit the other F, ND, on they project. Capillaries I throughout the insect's body cavity, figure 6. Insects have open circulatory systems and therefore cannot use bladder coulombic cavity L. Opening curly bracket dot dot. Y filtration to produce an excretory fluid. Metanephridium S. The cells of the Malpighian tubules actively transport uric acid, potassium ions, and sodium ions from the tissue fluid into the tubules. As these solutes are secreted into the tubules, water follows because of the difference in osmolarity. The walls of the Malpighian tubules have muscle cells that contract to help move the contents of the tubules toward the hindgut. Collecting nephridiopore. Tubules. The tubule fluid changes in composition while it is in the end hindgut. The contents of the hindgut are more acidic than nephrostome. The tubule fluid, as a result, uric acid becomes less. Coulombic fluid enters the Praduca ID and GK urine soluble and precipitates out of salt. Alon is it apprentice metanephridium fraud that is excreted through and enters the rectum. The epithelial cells of the hindgut and nephrostome, the nephridiopore, and rectum actively transport sodium and potassium ions from the gut contents back into the tissue fluid. Because 5 metanephridia in earthworms, the metanephridia of annelids are the uric acid molecules have precipitated out of solution. Arranged segmentally. The cross section at the left shows a pair of metanephridia. Longitudinal sections at the right show only one metanephrid. Water is free to follow the resorbed salts back into the of the 2 in each segment. Tissue fluid through osmosis, remaining in the rectum, are crystals of uric acid mixed with undigested food. This. Each segment of the earthworm contains a pair of dry matter, is what the insect eliminates. The Malpighian metanephridia, singular, metanephridium, from the tubule system is a highly effective mechanism for 88 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Excreting nitrogenous wastes and some salts without giving up a significant fraction of the animal's precious water supply. Vertebrate excretory systems. The major excretory organ of vertebrates is the kidney. The functional unit of the kidney is the nephron. Each human kidney has about 1 million nephrons. All vertebrate kidneys contain nephrons, yet the kidneys of different species can contribute to water and salt balance in opposite ways. Both marine and terrestrial vertebrates must conserve water. Some paleontologists believe that the immediate ancestors of the vertebrates lived in fresh water. If that one's true, the nephron would have evolved as a structure to excrete excess water. Indeed, the nephron can filter large quantities of blood and conserve valuable solutes such as glucose, amino acids, and certain ions while excreting water and nitrogenous wastes. How, then, have vertebrates adapted to environments where water must be conserved and salts excreted? The answer to this question differs among vertebrate groups. Even among the marine fishes, the adaptations of the bony fishes are different from those of the cartilaginous fishes. Marine bony fishes. Marine bony fishes cannot produce urine that is more concentrated than their tissue fluid, but unlike most marine animals, they osmoregulate their tissue fluid at only 1/4 to 1/3 the osmolarity of seawater. They prevent excessive loss of water by producing very little urine. Marine bony fishes take in seawater with their food, which results in large salt loads. The fish handle these salt loads by simply not absorbing some ions such as Mg or SO percent from their guts and by actively excreting others, such as C, from the gill membranes and from the renal tubules. Nitrogenous wastes are lost as ammonia from the gill membranes. Cartilaginous fishes. Cartilaginous fishes are osmoconformers, but not ionic conformers. Unlike marine bony fishes, cartilaginous fishes convert nitrogenous wastes to urea and another compound called trimethylamine oxide, and they retain large amounts of these compounds in their tissue fluids. As a result, their tissue fluids have an osmolarity close to that of seawater, so they do not lose body water to the environment by osmosis. These species have adapted to concentration of urea in the body fluids that would be toxic to other vertebrates. Sharks and rays still have the problem of excreting the large amounts of salts they take in with their food. One adaptation to solve this problem is a knuckle secreting rectal gland. Amphibians. Most amphibians live in or near fresh water and stay in humid habitats when they venture from the water. Like freshwater fishes, most amphibian species produce large amounts of dilute urine and consort salts. Some amphibians, however, have adapted to habitats that require water conservation. Amphibians living in very dry terrestrial environments have reduced the permeability of their skin to water. Some secrete a waxy substance that they spread over the skin to waterproof it. Several species of frogs that live in arid regions of Australia burrow deep into the ground and remain there during long dry periods. They enter estivation, a state of very low metabolic activity and therefore low water turnover. When it rains, the frogs come out of estivation, feed, and reproduce. Their most interesting adaptation is an enormous urinary bladder. Before entering estivation, they fill the bladder with dilute urine, which can amount to 1/3 of their body weight. This dilute urine serves as a water reservoir that is gradually resorbed into the blood during the long period of estivation. Australian Aboriginal peoples dig up estivating frogs as an emergency source of drinking water. Reptiles. Reptiles occupy habitats ranging from aquatic to extremely hot and dry. Three major adaptations have freed the reptiles from maintaining the close association with water that is necessary for most amphibians. First, reptiles do not need fresh water to reproduce because they employ internal fertilization and lay eggs with shells that retard evaporated water loss. Second, they have scaly, dry skin that retards evaporated water loss. Third, they excrete nitrogenous wastes as uric acid solids, losing little water in the process. Birds. Birds have the same adaptations for water conservation that reptiles have: internal fertilization, shelled eggs, skin that retards water loss, and uric acid is a nitrogenous waste product. In addition, birds can produce urine that is more concentrated than their tissue fluids. The nephron is the functional unit of the kidney. To understand how the kidney can fulfill different functions in different animal groups, we need to understand the structure of a nephron and how its parts work together. Vertebrate nephrons generally have three main constituents. The dense ball of capillaries called the glomerulus filters a portion of the blood plasma. The renal tubules receive and modify the glomerular filtrate. The paratubular capillaries bring substances to and take substances away from the renal tubules. The glomerulus and paratubular capillaries are vascular structures closely associated with the renal tubule. Figure 7. The different ways in which these three elements can work together influence the amount and composition of the urine. Blood is filtered in the glomerulus. The vascular component of the nephron is unusual in that the two capillary beds, the glomerulus and the paratubular capillaries, tilly in series between the arteriole that supplies them and the venule that drains them. The glomerulus plural, glomeruli, is a dense knot of very permeable vessels. Figure 8a. Blood enters the glomerulus through an afferent arteriole and exits. Institute for Advanced Studies, Jodhpur 89. Unit 7 System Physiology Animal. Through an efferent arteriole. The efferent arteriole gives rise to the paratubular capillaries, which surround the tubular component of the nephron. An afferent F4 to glomerulus, a not Bowman's arteriole supplies of capillaries, is a site capsule receives blood to be of blood filtration. HC0 in small molecules filtered from glomerular capillaries. Glomerulus. A tour of the nephron. These scanning electron micro-RFs stems show the anatomical basis for kidney function. Site of the blood vessels of the kidney, shown without the tubule components. Each glomerulus GI has an afferent R and an efferent filter T. Arteriole TH. Paratubular capillaries point are looser networks that surround the tubules of the nephron. B. The podocytes po. Tration or indirect contact with the capillaries of the glomerulus. Each podocyte has hundreds of tiny finger-like projections that create glomerulus filtration slits between them. Anything passing from the capillaries into the tubule of the nephron must pass through these slits. 0. A cross-section of the glomerulus surrounded by the tubule cells that form Bowman's capsule BC, which collects the filtrate in. Funnels it into the tubule T of the nephron. Also smaller than the fenestrations of the narrow slits between the finger-like projections of the podocytes. As a result of these anatomical adaptations, water and small molecules enter the renal tubule of the nephron. Figure 8 sense, but cells and proteins remain in the capillaries. Site of section in G5, NT, or phenolta. The glomerular filtration rate is high because TB glomerular capillary blood pressure is unusually high. Tubules that will be in because the capillaries of the glomerulus, along with secreted into the urine or covering of podocytes, are more permeable than and carry away other capillary beds in the body. Urine resorbed substances. Processing. The renal venule drains. The paratubular capillaries. The process filtrate urine of the individual nephrons enters collecting ducts and is delivered to a common duct leaving the kidney. Urine. 7. The vertebrate nephron. The vertebrate nephron consists of a renal tubule closely associated with two capillary beds, the glomerulus and the paratubular capillaries. The tubule component of the nephron, the renal tubule, begins with Bowman's capsule, which encloses the glomerulus. The glomerulus appears to be pushed into Bowman's capsule much like a fist pushed into an inflated balloon. The cells of the capsule that come into direct contact with the glomerular capillaries are called podocytes. Figure 8b. These highly specialized cells have numerous arm-like extensions, each with hundreds of fine, finger-like projections. The podocytes wrap around the capillaries so that their finger-like projections interdigitate and cover the capillaries completely. The glomerulus filters the blood to produce a fluid, the renal filtrate, that lacks cells and large molecules. The walls of the capillaries, the basal lamina of the capillary endothelium, and the podocytes of Bowman's capsule all participate in filtration. Fenestrations between the endothelial cells of the capillaries allow water and small molecules to leave them, but are too small to permit red blood cells to pass through. The meshwork of the basal lamina is even finer than the fenestrations, and it prevents large molecules from leaving the capillaries. The renal tubules convert glomerular filtrate to urine. The composition of the filtrate that first enters the nephron is similar to that of the blood plasma. This filtrate contains glucose, amino acids, ions, and nitrogenous wastes in the same concentrations as in the blood plasma, but it lacks the plasma proteins. As this fluid passes down the renal tubule, its composition changes as the cells of the tubule actively resorb certain molecules from the tubule fluid and secrete other molecules into it. When the tubule fluid leaves the kidney as urine, its composition is very different from that of the original filtrate. The function of the renal tubules is to control the composition of the urine by actively secreting and resorbing specific molecules. The blood in the paratubular capillaries brings to the renal tubules molecules that the tubule cells will secrete into the urine. It also carries away any molecules that have been resorbed from the urine. The mammalian excretory system. The adaptations of mammals and birds that allow them to produce urine hyperosmotic to their tissue fluid were an important step in vertebrate evolution. These adaptations enable the excretory system to conserve water while still excreting excess salts and nitrogenous wastes. Mammals and birds have high body temperatures and high metabolic rates, and therefore have the potential for a high rate of water loss. Being able to minimize water loss from their excretory systems made it possible for these highly active species to occupy arid habitats. 90 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Kidneys produce urine, which the bladder stores. We will use the excretory system of humans as our example of the mammalian excretory system. Humans have two kidneys just under the dorsal wall of the abdominal cavity in the mid-back region. Figure 9a. Each kidney filters blood, processes the filtrate into urine, and releases that urine into a duct called the ureter. The ureter of each kidney leads to the urinary bladder, where the urine is stored until it is excreted through the urethra. The urethra is a short tube that opens to the outside at the end of the penis in males or just anterior to the vaginal opening in females. At the kidneys are positioned in the copyright upper rear of the abdominal cavity. 1. Urinary bladder, urethra. Loop of Henle ascending limb. Loop of Henle descending limb. Vasa recta capillaries. Urine leaves the kidney from the inner surface of the medulla and is collected in the ureter. Bowman's capsule glomerulus. Two sphincter muscles surrounding the base of the urethra control the timing of urination. One of these sphincters is a smooth muscle and is controlled by the autonomic nervous system. As the bladder fills, stretch receptors in the walls of the bladder trigger a spinal reflex that relaxes this sphincter. This reflex is the only control of urination in infants. The other sphincter is a skeletal muscle and is controlled by the voluntary nervous system. When the bladder is very full, only deliberate conscious effort prevents urination. Afferent arteriole. Distal. Convoluted tubule. 9. The human excretory system of the human kidneys are positioned in the upper dorsal region of the abdominal cavity. B. The human kidney has a auricular internal tissue structure that is the basis for its function. Since the glomeruli and the proximal and distal convoluted tubules are located in the cortex of the kidney, but the loops of Henle run in parallel as straight sections. Down into the renal medulla and back up to the cortex. Collecting ducts run from the cortex to the inner surface of the medulla, where they open into the ureter. Nephrons have a regular arrangement in the kidney. The kidney is shaped like a kidney bean. When cut down, its long axis and split open as a bean splits open. Its important anatomical features are revealed figure 9b. The ureter and the renal artery and renal vein enter the kidney on its concave punched inside. The ureter extends into the kidney in several branches, the ends of which envelop kidney tissues called renal pyramids. The renal pyramids make up the internal core, or medulla, of the kidney. The medulla is surrounded by tissue with a different appearance, called the cortex. The renal artery and vein give rise to many arterioles and venules in the region between the cortex and the medulla. Each human kidney contains about 1 million nephrons, and their organization within the kidney is very regular. All of the glomeruli are located in the cortex. The initial segment of a renal tubule is called the proximal convoluted tubule. Proximal, because it is the first segment of renal tubule to receive the filtrate from the glomerulus, and convoluted, because it is twisted figure 9c. All the proximal convoluted tubules are also located in the cortex. At a certain point, the renal tubule takes a dive directly down into the medulla. The portion of the tubule in the medulla is the loop of Henle. It is called a loop because it runs straight down into the medulla, makes a hairpin turn, and rises straight back to the cortex. Not all nephrons have long loops of Henle, so-called cortical nephrons do not. Where the ascending limb of the loop of Henle reaches the cortex, it becomes the distal convoluted tubule. Distal, because it receives the filtrate from the glomerulus after the other renal tubule segments. The distal convoluted tubules of many nephrons join a common collecting duct in the cortex. The collecting ducts then run in parallel with the loops of Henle down through the renal pyramid. The collecting ducts. Institute for Advanced Studies, Jodhpur 91. Unit 7 System Physiology Animal. However, empty into the funnel-shaped pelvis, which narrows down to leave the kidney as the ureter. Blood vessels also have a regular arrangement in the kidney. The organization of the blood vessels of the kidney closely parallels the organization of the nephrons. See figure 9 cents. Smaller and smaller arteries branch from the renal artery and radiate into the cortex. Finally, an afferent arteriole carries blood to each glomerulus. Draining each glomerulus is an efferent arteriole that gives rise to the paratubular capillaries, most of which surround the cortical portions of the tubules. A few paratubular capillaries run into the medulla in parallel with the loops of Henle and the collecting ducts. These capillaries form the vasa recta. All the paratubular capillaries from a nephron join back together into a venule that joins with venules from other nephrons and eventually leads to the renal vein, which takes blood from the kidney. The volume of glomerular filtration is greater than the volume of urine. Most of the water and solutes filtered out of the glomerulus are resorbed and do not appear in the urine. We can reach this conclusion by comparing the rate of filtration by the glomeruli with the rate of urine production. The kidneys receive about 1 liter of blood per minute, or about 1,500 liters of blood per day. How much of this huge volume is filtered out of the glomeruli? The answer is about 12%. This is still a large volume, 180 liters per day. Since we normally urinate 2 to 3 liters per day, about 98% of the fluid volume that is filtered out of the glomerulus is returned to the blood. Where and how is this enormous fluid volume resorbed? Most filtrate is resorbed by the proximal convoluted tubule. The proximal convoluted tubule is responsible for most of the resorption of water and solutes from the glomerular filtrate. The cells of this section of the renal tubule are cuboidal, and their surfaces facing into the tubule have many microvilli, which increase their surface area for resorption. These cells have lots of mitochondria, an indication that they are metabolically active. They actively transport Na with C, following passively, and other solutes, such as glucose and amino acids, out of the tubule fluid. Almost all glucose and amino acid molecules that are filtered from the blood are actively resorbed by these cells and transported back into the tissue fluid. The active transport of solutes into the tissue fluid causes water to follow osmotically. The water and solutes moved into the tissue fluid are taken up by the paratubular capillaries and returned to the venous blood leaving the kidney. Despite the large volume of water and solutes resorbed by the proximal convoluted tubule, the overall osmolarity of the renal filtrate does not change. The fluid that enters the loop of Henle has the same osmolarity as the blood plasma, although its composition is different. How, then, does the kidney produce urine that is hypertonic to the blood plasma? The loop of Henle creates a concentration gradient in the surrounding tissue. Humans can produce urine that is 4 times more concentrated than their blood plasma. The vampire bat we encountered at the beginning of this chapter can produce urine that is 20 times more concentrated than its blood plasma, and some desert-dwelling animals, as we will see, can produce even greater concentrations. This concentrating ability of the mammalian kidney is due to the loops of Henle, which function as a countercurrent multiplier tilde system. The term countercurrent refers to the fact that the tubule fluid in the descending limb of the loop flows in the opposite direction from that in the ascending limb. Multiplier refers to the ability of this system to create a solute concentration gradient in the renal medulla. The loops of Henle do not themselves produce concentrated urine. Rather, they increase the osmolarity of the tissue fluid in the medulla in such a way that the tissue fluid concentration increases from the top to the bottom of the medulla. The segments of the loop of Henle differ anatomically and functionally. Cells of the descending limb and the initial cells of the ascending limb are flat, with no microvilli and few mitochondria. They are not specialized for transport. Partway up the ascending limb, the cells become specialized for active transport. These cells are cuboidal and have lots of mitochondria. Accordingly, the loop of Henle is divided into the thin descending limb, the thin ascending limb, and the thick ascending limb. To understand the countercurrent multiplier mechanism, it one's easiest to move backward through the renal tubule, starting with the thick ascending limb, figure 10. The thick ascending limb actively resorbs Cl, with Na following passively from the tubule fluid and moves it into the surrounding tissue fluid. The thick ascending limb is not permeable to water, so the resorption of Na and C raises the concentration of those solutes in the surrounding tissue fluid. The thin descending limb, in contrast, is rather permeable to water, but not very permeable to Na and C. Since the surrounding tissue fluid has been made more concentrated by the Na and C resorbed from the neighboring thick ascending limb, water once withdrawn osmotically from the tubule fluid in the descending limb. Therefore, the fluid in the descending limb becomes more and more concentrated as it flows toward the hairpin turn at the bottom of the renal medulla. The thin ascending limb, like the thick ascending limb, is not permeable to water. It is, however, permeable to Na and Ci. As the concentrated tubule fluid flows up the thin ascending limb, it is more concentrated than the surrounding tissue fluid, so Na and C diffuse out of it. When the tubule fluid reaches the thick ascending limb, active transport continues to move Na and C from the tubule fluid to the tissue fluid. As a result of the process described above, the tubule fluid reaching the distal convoluted tubule is less concentrated than the blood plasma, and the solutes that have been left behind in the renal medulla have created a concentration gradient in the surrounding tissue fluid. 92 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. The tissue fluid of the renal medulla becomes more and more concentrated as we move from the border with the cortex down to the tips of the renal pyramids. You may wonder why the blood flow through the medulla does not wash out the concentration gradient established by the loops of Henle. This is the significance of the parallel arrangement of the descending and ascending paratubular capillaries, the vasa recta, in the medulla. Countercurrent exchange between the descending and ascending vessels facilitates preservation of the concentration gradient. Filtrate in proximal. Blood plasma Bowman's convoluted distal convoluted. 300 mosum 1 capsule tubule glomerulus. 300 glucose amino acids and other 300 valuable solutes. Underscore. Blood il 200 light thick segment NT ascending limb in 300 NACL pumps NACL out of the urine and into the tissue. 150 H O fluid, but H O cannot follow because this region blood 1 O of H O of the tubule is impermeable to water. Out send pumping of NACL from the thick 350 NACL 8300 ascending limb sets up a concentration gradient. W 400 tilde 1 in the renal medulla. E A Ho 200 500 outer. O, GDO equals 1. W increased concentration of NACL in the tissue fine medulla 1400 E NACL 5400 equals fluid causes osmotic absorption of water from OE60 to descending limb. Thus sends intradendian 600 H, 0 S400, GE tubule fluid that enters the ascendal NIMGB. E equals G1 who, 2I. Percent 700 B, Saint, 600 E NACL LO0 feed IEOO, YI urine entering the collecting duct is less. FL reconcentrated in the tissue fluid, so is urine. 800 H, 0 equals, I 800 passes down the collecting to its CLTOsis water ELHO to the tissue FLUED and becomes near and more equals to I concentrated. Equals 900,800,800. And NACL equals, urea equals 1, tilde percent, 1.000 high 0 equals, 1100 water resorbed from the descending limb K and G. NACL less than underscore J ho the collecting duct leaves the medulla in the G 1100 T and 000 LR 0 1000 vasa recta. Dot. Medulla urea. The lower collecting duct is permeable to urea 1210 as well as to water. Urea is very concentrated. HEO underscore I. Zidone in the urine at this point, so it diffuses into the L tissue fluid. Some urea and TTD is arsine S in G loop of Henle and is recycled. Equals diffusion acti Ve. Transport L. Urine. 10 concentrating the urine a countercurrent multiplier mechanism enables the kidney to produce urine that is far more constant. Traded in the mammalian blood plasma. Water resorption begins in the distal convoluted tubule. The first portion of the distal convoluted tubule has properties similar to the thick ascending limb of the Luca panel. Na and C are transported out of the tubule. Fluid and water cannot follow. As a result, the tubule fluid becomes even more dilute. The later sections of the distal convoluted tubule, however, can be permeable to water, and water can be osmotically drawn into the surrounding tissue fluid. As the tubule fluid flows from the distal tubule to the collecting duct, it can be below or equal to the osmolarity of the blood plasma. Urine is concentrated in the collecting duct. The tubule fluid entering the collecting duct is at the same solute concentration as the blood plasma, but its composition is considerably different from that of the plasma. The major solute in the tubule is now urea, since salts were resorbed earlier in the nephron. As the tubule fluid flows down the collecting duct, it loses water osmotically to the surrounding tissue fluid. The concentration gradient established in the renal medulla by the loops of Henle creates the osmotic potential that withdraws water from the collecting ducts. The collecting ducts begin in the renal cortex and run through the renal medulla before emptying into the ureter at the tips of the renal pyramids. As the solute concentration of the surrounding tissue fluid increases, more and more water can be absorbed from the urine in the collecting duct. By the time it reaches the ureter, the urine can become greatly concentrated, with urea as a major solute. As water is withdrawn from the collecting duct, some urea also leaks out into the medullary tissue, adding to its osmotic potential. This urea diffuses back into the loop of Henle and is returned to the collecting duct. The recycling of urea in the renal medulla contributes significantly to the ability of the kidney to concentrate. Institute for Advanced Studies, Job for 93. Unit 7 System Physiology Animal. The urine in the collecting duct. Overall, the ability of a mammal to concentrate its urine is determined by the maximum concentration gradient it can establish in its renal medulla. An important adaptation for increasing the concentration gradient is to increase the lengths of the loops of Henle. The desert gerbil, for example, has such extremely long loops of Henle that its renal pyramid, each of its kidneys has only one. In contrast to ours, extends far out of the concave surface of the kidney, figure 11. These animals are so effective in conserving sodium ions, Na, and bicarbonate ions, HCOI, are filtered in the glomerulus. Renal tubule cells secrete HN. Exchange Fe or Na. Renal tubule ties lumen. Na. Glomerulus HCO5 plus high equals high E nat water THGT. They can survive on the water urea, ed by the K greater than H4. Metabolism of their food. They do not need to drink. Renal, R, WTEHCO, S, IHCOBCO. Closing square bracket. CO is converted back to HCO3 in renal tubule cell and transported back into the tissue fluid. CO is formed by the reaction of H goes H and diffuses into the tubule cell. 12. The kidney excretes acids and conserves bases. Bicarbonate ions are filtered out of the glomerulus, and renal tubule cells secrete hydrogen ions into the tubule fluid. In the renal tubule, the filtered bicarbonate buffers the secreted hydrogen ions and keeps the urine from becoming too acidic. The CO, formed by the reaction of bicarbonate and 11, the ability to concentrate the ability of the mammalian hydrogen ions is converted back to bicarbonate by the renal tubule cells and transported kidney to concentrate urine depends on the lengths of its loops of Henle relative to the overall size of the kidney. The kidney shown here from a desert gerbil has a single renal pyramid with loops of Henle so long that they protrude out of the medulla and into the ureter. The kidneys help regulate acid base balance. Besides regulating salt and water balance and excreting nitrogenous wastes, the kidneys have another important role. They regulate the hydrogen ion concentration, the pH of the blood. Blood pH is a critical variable because it influences the structure and therefore the function of proteins. One way to minimize pH changes in a chemical solution is to add a buffer, a substance that can either absorb or release hydrogen ions. The major buffers in the blood are bicarbonate ions, HCOJY, that are formed from the dissociation of carbonic acid, which in turn is formed by the hydration of CO, according to the following equilibrium reaction. CO plus H, O equals H, CO equals H plus HCO. Tilde you can see that if excess H plus ions are added to this reaction mixture, the reaction will move to the left and absorb the excess H. On the other hand, if H ions are removed from the reaction mixture, the reaction will move to the right and supply more H ions. The HCO tilde buffer system is important for controlling the pH of the blood because the reaction can be pushed to the right and pulled to the left physiologically. The lungs control the levels of CO in the blood, thus altering the acid portion of the reaction. The kidneys control the base portion of the reaction by altering the levels of H and HCO, trademark ions in the blood. The renal tubules secrete H into the tubule fluid and resorb HCO, trademark figure 12. The kidney has other buffering systems as well, and together they greatly enhance the ability of the kidney to eliminate acid from the blood back into the tissue fluid. Regulation of kidney functions. Several regulatory mechanisms act on the kidneys to maintain blood osmolarity and blood pressure. We will discuss these mechanisms separately, but keep in mind that they are always working together. The kidneys act to maintain the glomerular filtration rate. If the kidneys stop filtering blood, they cannot accomplish any of their functions. The maintenance of a constant glomerular filtration rate (GFR) depends on an adequate blood supply to the kidneys and an adequate blood pressure. Therefore, the kidneys have mechanisms to maintain their blood supply and blood pressure regardless of what is happening elsewhere in the body. Because these adaptations of the kidney support the maintenance of kidney function, they are called autoregulatory mechanisms. The kidney's autoregulatory adjustments compensate for decreases in cardiac output or decreases in blood pressure so that the GFR remains constant. Figure 13. One autoregulatory mechanism is the dilation expansion of the afferent renal arterioles when blood pressure falls. This dilation decreases the resistance in the arterioles and helps maintain blood pressure in the glomerular capillaries. If arterial dilation does not keep the GFR from falling, the kidney releases an enzyme, renin, into the blood. Renin acts on a circulating protein to convert it into an active hormone called angiotensin. Angiotensin has several effects that help restore the GFR to normal. First, angiotensin constricts the afferent renal arterioles, which elevates blood pressure in the glomerular capillaries. Second, it constricts peripheral blood vessels all over the body, an action that elevates central blood pressure. Third, it stimulates the adrenal Unit 7 System Physiology Animal. Cortex to release the hormone aldosterone. Aldosterone stimulates sodium resorption by the kidney, thereby making its resorption of water more effective. Enhanced water resorption helps maintain blood volume and therefore central blood pressure. Finally, angiotensin acts on the brain to stimulate thirst. Increased water intake in response to thirst increases blood volume and blood pressure. J. 150. T kidney failure. E normal kidney function. F count OKI. The GFR is nearly independent of 5 phi arterial pressure over a wide range. EZ equals E. When arterial pressure falls too low. E511 the kidney fails to produce urine. LA. L. O1 1IL. O50 100 150 200. Arterial pressure (mHg). 13. Maintaining the glomerular filtration rate. Glomerular filtration is driven by arterial pressure, but autoregulatory mechanisms prevent rises and falls in glomerular filtration rate (GFR) over a wide range of pressures. Blood pressure and osmolarity are regulated by ADH. When you lose blood volume, your blood pressure tends to fall. Besides activating the kidney autoregulatory mechanisms described in the previous section, a drop in blood pressure decreases the activity of the stretch receptors in the walls of the aorta and the carotid arteries. These stretch receptors provide information to cells in the hypothalamus that produce antidiuretic hormone (ADH), also called vasopressin, and send it down their axons to the posterior pituitary gland. As stretch receptor activity decreases, the production and release of this hormone increases. Figure 14. ADH acts on the distal convoluted tubules and collecting ducts of the kidney to increase their permeability to water. When the circulating level of ADH is high, the distal tubules and the collecting ducts are very permeable to water. More water is resorbed from the urine, and only small quantities of concentrated urine are produced, thus conserving blood volume and maintaining blood pressure. When ADH levels are low, water is not resorbed from the collecting ducts, and lots of dilute urine is produced. ADH controls the permeability of the collecting ducts by stimulating the production and activity of membrane proteins that form water channels. These proteins, called aquaporins, are found in many tissues that are permeable to water, for example, the capillary, endothelium, red blood cells, and the proximal convoluted tubules of the kidney. Differences among tissues in water permeability can be related to the presence or absence of aquaporins. Aquaporins are expressed in the descending limb of the loop of Henle, for example, but not in the ascending limb. One particular aquaporin that is found in distal convoluted tubule and collecting duct cells is controlled by ADH on both the long-term and a short-term basis. Over the long term, ADH levels influence the expression of the gene for this aquaporin. Over the short term, ADH controls the insertion of the aquaporin into the cell's plasma membranes. ADH also helps regulate blood osmolarity by controlling water resorption. Sensory cells in the hypothalamus monitor the osmolarity of the blood. If blood osmolarity increases, these osmoreceptors stimulate increased release of ADH to enhance water resorption from the kidneys. The osmoreceptors also stimulate thirst. The resulting water retention and water intake dilutes the blood as it expands blood volume. Regulation of blood osmolarity, blood pressure. Rise in. Blood osmolarity pressure. Osmoreceptors stretch receptors detect increases in molarity and blood P stimulate ADH and inhibit ADH release F tilde T. I posterior T renal excretion of pituitary constriction of solutes and resorption peripheral blood. A PO decreased vessels elevates blood osmolarity, blood pressure. In the kidney, plus in the periphery, ADH increases ADH causes permeability of 4 well of ADH blood vessels collecting duct in bloodstream to constrict cells in distal. Tubule cells to DO. Resorption of water P helps maintain blood volume. 14 antidiuretic hormone increases blood pressure and promotes water resorption. ADH is produced by neurons in the hypothalamus and released from their axons in the posterior pituitary. The release of ADH is stimulated by hypothalamic osmoreceptors and inhibited by stretch receptors in the great arteries. The heart produces a hormone that influences kidney function. When systemic venous return to the heart increases and the atria of the heart become more stretched, the atrial muscle fibers release a peptide hormone called atrial natriuretic peptide ANP. This peptide hormone enters the circulation, and when it reaches the kidney, it decreases the resorption of sodium. The result is an increased loss of sodium and water, which has the effect of lowering blood volume and blood pressure. In pathological situations in which a weakened heart cannot pump enough blood to keep up with the venous return, congestive heart failure, the atria becomes stretched more than normal, and the resulting high levels of ANP in the blood can be a useful diagnostic measure. Institute for Advanced Studies, Jodhpur 95. Unit 7 System Physiology Animal. G thermoregulation, comfort zone, body temperature, physical, chemical, neural regulation, acclimatization. Homeostasis and temperature on the body surface of an animal. This transfer is always regulation from an area of higher temperature to one of lower. The temperature of a living cell affects the rate of its metabolic processes. An animal can grow faster and respond to the environment more rapidly if its cells are kept warm. In fact, zoologists believe that the ability of some higher animals to maintain a constant homeostatic, relatively high body temperature is a major reason for their evolutionary success. This ability to control the temperature of the body is called thermoregulation, heat control, and involves the nervous, endocrine, respiratory, and circulatory systems in higher animals. The impact of temperature on animal life. Every animal's physiological functions are inexorably linked to temperature, because metabolism is sensitive to changes in internal temperature. Thus, temperature has been a strong source of selective pressure on all animals. The rate of cellular respiration increases with temperature up to a point. When the temperature rises above the temperature optima at which enzymes most efficiently catalyze their chemical reactions, the rates decline as the enzymes begin to denature. The chemical interactions holding the enzymes in their three-dimensional shape are also disrupted. Thus, the result enzyme evolution have frequently been enzymes with temperature optima that reflect an animal's habitat. For example, a digestive enzyme in a trout might function optimally at 10 degrees. For example, a digestive enzyme in a trout might function optimally at 10 degrees Celsius, whereas another enzyme in the human body that catalyzes the same reaction functions best at 37 degrees Celsius. Higher temperatures cause the proteins and nucleic acids to denature, and lower temperature because heat. For example, a digestive enzyme in a trout might function optimally at 10 degrees Celsius, whereas another enzyme in the human body that catalyzes the same reaction functions best at 37 degrees Celsius. Higher temperatures cause the proteins and nucleic acids to denature, and lower temperature because heat moves down thermal gradients. For example, when you sit on the cold ground, you lose heat, and when you sit on warm sand, you gain heat. Convection is the movement of air or liquid over the surface of a body. It contributes to heat loss if the air is cooler than the body, or heat gain if the air is warmer than the body. On a cool day, your body loses heat by convection because your skin temperature is higher than the surrounding air temperature. Equals heat gain and loss for a terrestrial reptile in a typical terrestrial environment. Heat is either gained or lost by objects in direct contact with the animal: conduction, by air currents; convection, by exhaled air evaporation, or by electromagnetic waves, embiation. Temperatures may cause membranes to change from a fluid to a solid state, which can interfere with many cellular processes, such as active transport pumps. Animals can guard against these damaging effects of temperature fluctuations by balancing heat gains and heat losses with their environment. Heat gains and losses. Animals produce heat as a byproduct of metabolism and either gain heat from or lose it to the environment. The total body temperature is a result of an interaction of these factors and can be expressed as body temperature equals heat produced metabolically plus heat gain from the environment, heat loss to the environment. Animals use four physical processes to exchange heat with the environment: conduction, convection, evaporation, and radiation. Figure 1. Conduction is the direct transfer of thermal motion heat between molecules of the environment, and those evaporation is loss of heat from a surface's water molecules escape in the form of a gas. It once useful only to terrestrial animals. For example, humans and some other mammals, chimpanzees and horses, have sweat glands that actively move watery solutions through pores to the skin surface. When skin temperature is high, water at the surface absorbs enough thermal energy to break the hydrogen bonds holding the individual water molecules together, and they depart from the surface, carrying heat with them. As long as the environmental humidity is low enough to permit complete evaporation, sweating can rid the mammalian body of excess heat. However, the water must evaporate. Sweat dripping from a mammal has no cooling effect at all. Radiation is the emission of electromagnetic waves that objects, such as another animal's body or the sun, produce. Radiation can transfer heat between objects that are not in direct contact with each other, as happens when an animal suns itself. 96 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Temperature and life. Temperatures vary enormously over the face of Earth, from the boiling hot springs of Yellowstone National Park to the interior of Antarctica, where the temperature can fall below 80 degrees Celsius. Because heat always moves from a warmer to a cooler object, any change in the temperature of the environment causes a change in the temperature of an organism in that environment, unless the organism does something to regulate its temperature. Living cells can function over only a narrow range of temperatures. If cells cool to below 0 degrees Celsius, ice crystals damage their structures. Some animals have adaptations, such as antifreeze molecules in their blood, that help them resist freezing. Others have adaptations that enable them to survive freezing. Generally, however, cells must remain above 0 degrees Celsius to stay alive. The upper temperature limit is less than 45 degrees Celsius for most cells. Some specialized algae can grow in hot springs at 70 degrees Celsius, and some archaea can live at near 100 degrees Celsius, but in general, proteins begin to denature and lose their function as temperatures approach 45 degrees Celsius. Therefore, most cellular functions are limited to the range between 0 degrees Celsius and 45 degrees Celsius, which are considered the thermal limits for life. A particular species, however, generally has much narrower limits. Co is a measure of temperature sensitivity. Even within the range from 0 degrees to 45 degrees Celsius, changes in tissue temperature create problems for animals. Most physiological processes, like the biochemical reactions that most physiological processes, like the biochemical reactions that constitute them, are temperature sensitive, going faster at higher temperatures. See figure. The temperature sensitivity of a reaction or process can be described in terms of QP9, a quotient calculated by dividing the rate of a process or reaction at a certain temperature, Ri, by the rate of that process or reaction at a temperature 10 degrees Celsius lower, Ri underscore cent. Q1 can be measured for a simple enzymatic reaction or for a complex physiological process, such as rate of oxygen consumption. If a reaction or process is not temperature sensitive, it has a co of 1. Most biological co-values are between 2 and 3. A Q of 2 means that the reaction rate doubles as temperature increases by 10 degrees Celsius, and a co of 3 indicates a tripling of the rate. Figure. Changes in tissue temperature can be particularly disruptive to an animal's functioning because not all of its component reactions have the same Q. Reactions with different co-values are linked together in complex networks that carry out physiological processes. Changes in tissue temperature shift the rates of some reactions more than those of others, thus disrupting the balance and integration that the processes require. To maintain homeostasis, organisms must be able to compensate for or prevent changes in temperature. 10 Co equals 7 R7 R0. This reaction rate triples with each 10 degrees Celsius rise in temperature. Kip equals 3 F. O1 O equals 2. This reaction rate doubles with each 10 degrees Celsius rise in temperature. The rates of most within this range biochemical reactions and physiological processes fall. R8A. OF.